Trypanosomes are microscopic, single-celled parasites that belong to the genus Trypanosoma. These organisms are best known for causing serious diseases in humans and animals, most notably African sleeping sickness and Chagas disease. African trypanosomiasis occurs in rural areas in the tsetse fly areas of sub-Saharan Africa. Types of transmission of trypanosomes. Transmission of trypanosomes by the flies can be mechanical or cyclic, and salivarian or stercorarian. Cyclical transmission of the parasite is transmission through a vector in which the parasite undergoes developmental changes, and multiplication inside the vector before being passed on to a new host. Mechanical transmission. This is transmission of a parasite by a vector without undergoing development or multiplication inside the vector. The vector acts only as a mechanical carrier. Transmission occurs quickly, often by contamination of mouthparts. The flies bite an infected animal and carry parasites from an infected animal's blood on their mouthparts to the next host. Mechanical transmission can also occur through blood transfusion of fresh blood. Salivarian transmission. This occurs when the infective stage of the parasite is injected into the host through the saliva of the insect vector during a blood meal. Stercorarian transmission. This transmission occurs when the infective stage is passed in the feces of the insect vector, and infection happens when the parasite-containing feces contaminate the bite wound, mucous membranes, or conjunctiva. Cyclical transmission. This is where the trypanosomes undergo a cycle of development and multiplication in digestive tract of the fly until the infective metacyclic trypanosomes are produced. African trypanosomiasis is caused by Trypanosoma brucei gambiense and Trypanosoma brucei rhodesiense. Trypanosoma brucei gambiense is found in West Africa and Western Central Africa, extending from Senegal across to Sudan and down to Angola. Trypanosoma brucei rhodesiense is found in East Africa, Central Africa, and Southern Africa, extending from Ethiopia down to Botswana. Note, in Uganda, both species occur, TB gambiense in the northwest and TB rhodesiense in the southeast. Their impact on global health makes them a subject of intense scientific study. Among the many species of trypanosomes, two are especially significant for human health. Trypanosoma brucei is responsible for African sleeping sickness, also known as African trypanosomiasis, which is transmitted by the tsetse fly and primarily affects people in sub-Saharan Africa. Trypanosoma cruzi, on the other hand, causes Chagas disease in Central and South America, spread by triatomine bugs. Both diseases can be fatal if left untreated. The life cycle of trypanosomes is complex, involving both insect vectors and mammalian hosts. Life cycle, transmission metacyclic. Trypomastigotes are injected through skin when Glossina fly takes a blood meal. The parasites develop into long, slender trypomastigotes, which multiply at the site of inoculation and later in the blood, lymphatic system, and tissue fluid. They are carried to the heart and various organs of the body, and in the later stages of infection, they invade the central nervous system, CNS. Trypomastigotes are ingested by a Glossina fly when it sucks blood. In the midgut of the fly, the parasites develop and multiply. After 2-3 weeks, the trypomastigotes migrate to the salivary glands of the tsetse fly, where they become epimastigotes. They multiply and develop into infective metacyclic trypomastigotes. Diagnosing trypanosome infections involves several laboratory techniques. Microscopic examination of blood, lymph, or cerebral spinal fluid samples can reveal the parasites. Concentration techniques such as Strout techniques, microhematocrit techniques, and triple centrifugation concentration technique. Serological tests detect antibodies or antigens specific to trypanosomes. Molecular methods, such as polymerase chain reaction, or PCR, offer high sensitivity and specificity, aiding in early and accurate diagnosis. Differences between Gambiense and Rhodesiense trypanosomiasis. West African trypanosomiasis is found in West and Central Africa, particularly in riverine areas, whereas East African trypanosomiasis is confined to East and Southern Africa, mainly in open savanna regions. The reservoirs of infection also differ. Humans serve as the primary reservoir for TB gambiense, whereas wild animals and cattle act as reservoirs for TB rhodesiense, making the latter more of a zoonotic disease. West African trypanosomiasis is transmitted by riverine Glossina flies, whereas East African trypanosomiasis is transmitted by woodland Glossina flies. West African trypanosomiasis parasitemia is low, whereas East African trypanosomiasis parasitemia is high. West African trypanosomiasis progresses slowly, often taking months or years before severe symptoms appear, whereas East African trypanosomiasis develops rapidly, sometimes within weeks. Patients with TB gambiense infection typically present with intermittent fever, lymphadenopathy, headaches, and itching. A characteristic feature is Winterbottom's sign, posterior cervical lymph node enlargement. In contrast, TB rhodesiense infection often begins with a painful chancre at the bite site, followed by high fever, muscle pains, and early involvement of the central nervous system. Thank you for watching. Subscribe and share.